studying the certain Sagocloson sea slugs and how they become photosynthetic when they eat macroalgae and they steal the chloroplasts from the algae and incorporate them inside their own cells as photosynthetically active kleptoplasts. We were specifically focusing on studying the light acclimation mechanisms and how they differ between the algae that the slugs eat and the slugs themselves after they incorporate the chloroplasts inside their own cells. And we were specifically focusing on a mechanism called state transitions, which green algae use to balance light energy in changing light conditions. Surprisingly, we found that one specific entire group of green algae called Bryopsidales have lost state transitions, which is quite strange because state transitions are very common in green algae. And in line with that, most of the algae that were eaten by the slugs did contain strong state transitions, but the state transitions were immediately lost when the chloroplasts were incorporated by the slugs. We then found that in the slugs, the chloroplasts or kleptoplasts are of very spherical shape, whereas in the algae they are of very diverse shapes and sizes. And that means that there are structural changes going on during the incorporation process as kleptoplasts into the slugs. We were able to update our knowledge concerning the light acclimation mechanisms of the different green algae and also the photosynthetic sea slugs that eat those green algae quite significantly. Finding out that there are these huge structural changes going on during the incorporation process as kleptoplasts, we are giving a mechanistic explanation of how these state transitions are lost during the incorporation process into the slugs.